So welcome everyone. My name is Anna Maria Niewidomska, and I'm the Global Market Segment Manager for Public Health at Oxford Nanopore Technologies. I'm delighted to welcome you all today to this knowledge exchange, where we'll be looking at how Oxford Nanopore Technologies can provide a rapid and comprehensive metagenomic approach for characterizing bacteria, fungi, and viruses directly from respiratory samples. After the talks, we'll have a live Q&A session, so please do add your questions to the Q&A box, which you can find at the bottom of the screen. You can also upvote the questions that you would most like answered, and we'll try to get through as many as we can today. This knowledge exchange is being recorded and will be made available on demand after the broadcast. So let's jump into it. When we talk about metagenomics, there are quite a few things that come to people's minds. So I thought it would be useful today to just discuss the differences between the different solutions that Oxford Nanopore can offer for metagenomic and metagenomic-like sequencing. So one of the first is 16S ribosomal RNA sequencing. And while people often consider this alongside metagenomics, it's not truly metagenomic sequencing. It's a targeted sequencing of a conserved region that's shared amongst many bacteria, which is the 16S ribosomal RNA gene. And this is typically done as a fast and cost-effective way to get a general idea of bacterial species ID in a community, to get an idea of the community profile, and this has applications in both food and environmental testing. Pathogen surveillance, on the other hand, tends to be a method for detecting specific infectious disease pathogens, typically those that cause disease to some species that we're interested in a truly agnostic way. And ideally would be able to capture both viruses, bacteria, and fungi. Ideally, you would also be able to pull out antimicrobial resistance profiles from this, and it would be used to monitor animal and environmental health for biosecurity purposes and biosurveillance. Microbiome metagenomic sequencing, on the other hand, typically is used for more research purposes in basic research, again, to get an idea of microbial diversity. But in this case, we're not just looking for pathogenic microbes. It's just to get an idea of the entire microbial community. And most people's goal is to assemble MAGs, or metagenome assemble genomes, out of these species. And of course, this often has applications in looking at the microbiome's role in health and its associations with certain diseases. However, the focus of today's talk will be for pathogen metagenomic surveillance. So it can be very confusing to figure out which kit you need for which application. So I thought I would summarize them in this table so that it's quite easy for you depending on which application you want to pursue. So with pathogen surveillance, we really don't need to have any prior taxonomic knowledge. So there's no primers involved. You don't need to sequence a conserved region. You're just sequencing everything that's in your sample. This is ideal for detecting co-infections, divergent or novel viruses, because again, you don't need to have any prior knowledge of what's in your sample for this. And it's also able to, because of its dual arm protocol, it's able to capture both viral and acellular pathogens that might be in one specific arm, as well as bacterial and fungal pathogens in the other arm. The data complexity tends to be a little bit higher than 16S ribosomal RNA sequencing, but not quite as high as complete metagenomic sequencing. We do have an extraction protocol that's included in this end-to-end protocol that can be found on our website. The kit that's used for this protocol is the rapid PCR barcoding kit. It does require some third-party reagents, and the protocol includes amplification or a reverse transcription and PCR step. So this means that you won't be able to detect methylation. And when you're done with your library prep, you can run this on a MinION flow cell, which is compatible with either a MinION or a GridION device. And if you just want to get a quick idea of what's in your sample, you can use our graphical user interface data analysis software, our Epidemy software, and run our workflow for metagenomics in that pipeline, or choose your own workflow. And with that, I'd now like to welcome Alex Trotter, who's a development scientist in our translational development department at Oxford Nanopore Technologies, to take us through the process. Over to you, Alex.
Thanks, Anna. Hi, everyone. I'm Alex from the Translational Development team here at Oxford Nanopore, and I'm going to walk you through the wet lab section now and a bit of the logic behind why we're doing any of this to begin with, okay? So, surveillance metagenomics. What is that? What are we talking about here? So, we're looking here for an approach that's comprehensive. We want to analyze everything. We want to see all the genetic material that's in our sample, whether that be DNA, whether it be RNA. We want to see it all. We want to see it all in one application, okay? How are we going to do that? We're going to take an agnostic approach to this, and this is really important because that means we'll be able to capture everything here in a non-targeted way. We don't want to target specific things. We don't want to introduce any bias to what we might see. We want to see it all. We don't want to see what we're expecting to see, right? We want to be able to find everything. We want to be able to find new things. We want to see what we might have been missing before, because as we know, what you're missing matters. And we're going to try and do this in an accurate way, right? We want to supersede those slow and insensitive culture methods that have been dominating this space for so long. We want to remove them because they're slow, and we want to remove them because they miss things sometimes. We want to be able to sequence everything that's in there. We're not just looking for what we've managed to culture, what we've managed to grow because we've given it the right medium, we've given it the right selection pressure. Maybe it doesn't even want to grow at all. This way, we want to see everything that's in a sample so we know exactly what is there. So, why are we going to do this? Why? That's great to find everything, but what's the point? What are we looking for? Why do we want to do this? Well, a few reasons. Pathogen identification, okay? We want to be able to identify everything. That's the simplest line here. We want to be able to find all the bacteria, all the fungi, all the viruses. We want to be able to find what's there. And once we've found it, we want to learn a bit more about it. If we've got its genome, we can look and see what sort of antimicrobial resistance genes are there. We can then use this for genomic epidemiology. We can track outbreaks with this. We can see what is going where, how different is it when it moves. We can identify new mutations, new variants, new everything. We can see it as it happens. We don't have to know what it already looked like to be able to look at it now. And then we can also do other things, virome studies. We can look at how things are interacting with their ecosystem, with their surroundings, and where they are, okay? Now, why would you want to use Oxford Nanopore for that? That's the most important thing here now, right? So, the usual reasons. We're talking flexible, we're talking fast, and we're talking accessible. So, we're going to be flexible with our sample batching. The number of samples that we're putting on sequencing at one time, that's going to be flexible. So, if we're in a rush, if it's urgent, we can do it now. We don't have to wait till that sequencing run is on next week. We can go when we're ready. Likewise, if we're not ready yet, we can keep batching that up. We can go when is suitable for us. The sequencing, the analysis, the protocol, we want it all to be rapid, okay? We want that real-time access to data. As nanopore sequencing is running, the analysis can be running in real time. That gives us rapid turnaround from our sample to our result. And we can do this anywhere, right? So the platform's accessible. We've got simple workflows here. We've got simple and indeed very often small machines here to sequence on that can really empower any lab, any scale to be able to do this. We can get this out there and deploy it wherever it needs to be. So now we're going to look through that wet lab workflow. I'm going to walk you through all the steps, all the reasoning behind each of the steps as we go through. So here's an overview, right? This is surveillance metagenomics. Here's our pathway from sample to result. We're going to be splitting our sample in two, you'll see here. There's two paths here, two arms. It's a dual arm approach, and that's going to be really important for a few reasons that we'll discuss later. We'll split those apart, and then at the end, we're going to bring them back together and then sequence them with this rapid kit. It's the rapid PCR barcoding kit. It's a kit 14 chemistry kit. It's going on an R10 flow cell. Now, this split here can be really important, really useful for us, because if you're only interested in one kind of thing, you can just look at that. If you're only looking at bacteria, fungi, do that arm. If you're only interested in viral stuff, do that arm. If you're interested in everything, you can do both. It's up to you, and it will be flexible in that way. So let's look at where they're different here, right? So bacteria and fungi, we're splitting them off into one side because of the way the cells are in terms of how we're going to remove the host background. That's going to mean we're going to need a depletion, right? We're going to do host depletion using saponin and HL-SAN, and then we're going to move to that extraction. Meanwhile, the viral stuff, not using saponin because we don't have anything to lyse there, we're just going to be using that HL-SAN and then going into that extraction. Those extractions are the same. It's going to be using the MagMax viral pathogen kit, lovely little kit. It could be automated on something like a Kingfisher, for example. That's the same extraction for those. The only difference will be that we're using bead beating in the bacterial and fungal arm, which we're not using in the viral arm. That would just chop everything up more than we wanted it to. After that, the bacterial and fungal side is ready. For the viral side, we're going to need to do reverse transcription, right? So we're going to have a nice RT step, make some cDNA. We'll talk about that and how that works a bit later. But once those bits are done, the protocol's the same for both sides. Once we've got to that PCR, they're going in the same PCR all together. That's simple now. And after we've done that PCR, we've gone and quantified, then we're going to normalize, pull everything back together, and we're down to one tube. So a dual arm approach, why? Why? Well, most importantly, sensitivity. That's really what this is about, is being sensitive for everything. Trying to get a balance of sensitivity for bacteria, fungi, viral, all in one approach, really difficult. The best way we've found to do that is to separate these out while they go through this process at the start. So we're going to do a really hard centrifuge. That's going to split it out. We're going to get our fungal cells, our bacterial cells, and our human cells all down at the bottom. Our viral cells and our acellular stuff, that's going to stay at the top. We can take that out, separate it, and then we can do a depletion on that bacterial and fungal side to get rid of all that human. So this is just the best way for us to get that coverage, right? So where have we got this from? Like, what is this based on? So the principles behind this, if you are interested in where these came from and how they really, really work, come from these two places, okay? So the bacterial and fungal, that cellular side, that is from this paper by Justin O'Grady's group, which brought in this saponin host cell depletion method, and it gets rid of at least 1,000-fold reduction in host nucleic acid, and that's from respiratory samples, for example. Can be even higher. If you're using other sample types such as blood, that could be even higher than that. But we're looking at getting that much out. We need to really, really reduce the amount of human background in there, because otherwise we're talking about looking for a needle in a haystack, right? Then the size of a bacterial cell compared to the size of the genome, at least, of a human cell, it's a big difference. We're looking needle in a haystack. You then think about how many bacterial or fungal cells you might be finding in a sample that's taken compared to how many human samples in there. That needle's now a grain of sand in the ocean. So it's really hard to look for them without doing a really efficient and good high quality depletion. And on the other side, the viral and acellular arm, that comes from this original Smart9N method that came from Josh Quick's group. However, we have optimized it a little bit to improve detection and genome recovery of some short segmented viral genomes such as flu A. I'm going to talk about that much, much later in the talk, but we'll really dive into that when we do. But for now, let's just talk about Smart9 and what this is, right? So what on earth? What is Rapid Smart9N? Well, it's a template switching mechanism. Works at the 5' end of RNA template using 9 monomers. What's that? Well, we're going to be using this 9N primer, and what that is it's a sequencing adapter primer with 9 ends on it. Those ends are going to randomly bind to all the RNA in the sample. We've done that, now we have our RNA, but it's got 9N primers attached. We're going to use a Maxima H enzyme that's going to reverse transcribe RNA, working along from that 9N primer to the other end. At the other end, it's going to add 3 Cs, that's for template switching. TSO primer comes in with 3 Gs, binds that to the 3 Cs, and that switches the template around so that we can make that second strand of cDNA. And this enables us to do both single and double-stranded RNA viruses, right? So we then got those cDNA templates, then they're tagged. They're tagged with the sequencing adapter. Also, the adapter from that 9N primer is going to randomly insert into the DNA that was already in there if we had DNA viruses, for example. So now everything's got a sequencing adapter and it's ready to go into the rapid PCR barcoding kit. That's where we're going to be doing PCR to amplify this all up. So what are we talking about here? If you're a nanopore aficionado, now I'm probably talking your language and you start knowing where we are, right? Rapid PCR barcoding kit, RPB114. This is the sequencing kit we're going to use. It's a rapid one. It's using transposon complex to start amplification. We're using PCR to amplify everything up. This kit is simple, it's rapid, and it works with really low starting inputs. For example, from this application, the starting inputs are going to be less than a nanogram per microliter, so we're working from really low inputs. Now, I've said here that we can multiplex up to 11 samples. You might be looking at that and thinking, what? This is a 24 barcoded kit. Why are we doing 11? Well, remember, we're splitting each of those samples in two, right? So every sample is going to have two barcodes eventually. 11 would take us up to 22, leaving a little space for some controls. And I really do recommend putting at least one negative control or no template control, a blank, something in there. When we're doing a metagenomics approach like this, it's really important to see what the background in the sequencing result looks like, okay? So what are we going to need? Well, when it comes to our reagents for sequencing, we're going to need that rapid PCR barcoding kit, RPB, and then we're going to need a MinION flow cell, and that can either go on a MinION device or a GridION device. You will need to get some third-party reagents as well for the stuff that was upstream. The depletion, the extraction, the PCR all need some third-party reagents. Those will be listed for you at the start of the protocol, but this is the nanopore stuff we're sequencing with. And then once we've done that, we've got our sequencing on, it's on the device, it's analyzing in real time. Those default settings for barcoding for this kit are for 72 hours using high accuracy base calling, but you could run it for less, right? You're flexible to that. You could do your analysis earlier. You could take it off at 24 hours. You could look at it in real time when it comes to doing your analysis. Speaking of analysis, my colleague Natalia is going to now talk you through how to do all of this analysis. And over to you, Natalia.
Thank you very much, Alex. And now I'm going to go through how to perform the downstream analysis of pathogens surveillance. We recommend to use WF Metagenomics, which allows you the identification of bacterial, fungal, and viral pathogens in a blood sample. The workflow is available through the Epi2Me desktop application, but also you can run it through the command line, and it allows running multiple samples. The only thing you need to take care would be just to choose the specific settings for this use for pathogen surveillance. Workflow Metagenomics is a bioinformatics pipeline written in Nextflow, and it takes as input the output from MinNow. Base calling, we recommend to perform live base calling using the high accuracy model, and from the output of MinNow would be the input of WF Metagenomics. So we will need to use these FASTQ or BAM files, and then we can run the pipeline. The pipeline first performs a read filtering based on the quality of the reads and also the length, and then it uses a third-party tool such as Kraken to perform the taxonomic classification of the reads. And finally, it estimates the abundances of the different taxa by using Bracken. The main output is an HTML report that I'll show you later that contains some information about how the sequencing, how the read length distribution, the accuracy, but also the taxonomic profile of the samples. So how could we run WF Metagenomics? As I mentioned, we recommend to use the Epi2Me desktop application, which is an open analysis platform that can be installed in Windows, in Macintosh, in Linux, and then allows you to run the workflows on the device, for example, on the laptop, or in the cloud. And it contains a collection of different workflows that are curated and maintained by experts in long-read sequence analysis. So what we will need is just first to select the workflow, WF Metagenomics, and then we install the workflows. The app automatically checks that if you satisfy the minimum requirements to run the workflow or not. And then we recommend always to run the demo data, which is a test to make sure that everything is well installed and properly set it. So that will be our first step. Once we are sure that everything is working as expected, we can analyze our samples. And first, to do that, we will need to select our inputs. So we select the FASTQ or BAM files. Also, we recommend to use a sample seed and select the PF8 database. Additionally, we set the Kraken confidence score to 0.01, and then in this left panel you can see that there are different options. You can, for example, choose if you want to output and classify reads to perform model analysis with them, or if you want to have some files with the taxonomic assignment of each read. And once we have set up the analysis, we can launch the workflow. As I mentioned, I'm gonna show you how to do this because a pig sometimes is worthier than 1,000 words. So here we have the app. I'm running this in my laptop, which is Windows device. And we go to launch. We select Metagenomics. In this case, I already have it installed. As you can see here, first you have like a README with some information about the workflow, how it does, which are the frequently asked questions, in case you need more information. And in the right side, you'll see in options that, for example, if you need to check for updates, we always recommend to run the latest version of the workflow, and also to run the demo, as I mentioned before. So now we can set up the analysis by providing the FASTQ files, which in this case is some samples with different barcodes. And we recommend to use the sample seed option because it allows you to provide more meaningful names and they will appear in the report. So that will help you when analyzing the report. If we move to the reference options, we can select the database, which would be plus PF8 database. You can also provide a custom one if you need it. And in the Kraken 2 options, we set the confidence score threshold to 0.01. As I mentioned earlier, in the left panel, you can explore the rest of the options that are available. Now we can launch the workflow. So it will run some comprobations and it will start. Here in this screen, you can see the progress of the workflow, how it runs. And you'll see different progress bars of the different steps that are happening, so you can make sure when the workflow has finished. Okay, as I mentioned earlier, there is also another option, which is running the workflow through the command line, but in this case, you would need to install Nextflow, Java, and Docker on Singularity, and then when writing the command, you will need to provide the same options, the Kraken confidence and also the database. The workflow is available in our GitHub, as you can see here. As an additional options, there are other databases, although we recommend to use plus PF8 because it contains standards, which are archaea, bacterial, viral, and human sequences, but also protozoans and fungi. And it's capped at gigabytes, which allows you to run it in a laptop with 16 gigabytes of RAM memory. And if you want to perform the antimicrobial resistance profile of the sample, that's also possible, checking the corresponding option, and it will add a step when Abricate can scan reads against the database for those genes. As I mentioned earlier, the main report or the main output of the workflow is the HTML report. That can be viewed in the app or can be opened by anyone with a web browser, and it contains some basic sequencing information, like, for example, the quality distribution of the reads, or also the read length distribution. It also contains some plots to help you to explore the taxonomic composition of the sample, like, for example, this Sankey plot, in which you can select the rank, species, genus, family, a cutoff of abundance, and then see what is in the sample. And the main main output within the report would be the abundance table that allows you to identify for different taxa or pathogens in the different samples, and you can sort it by abundance. You can use the search bar to look for a specific taxon or export the table if you need it for other analysis. And if we explore the analysis that we have just set up, we'll see that now it says completed. In options, we can also rename the analysis and if we go, for example, to logs, we'll see the next flow log that contains some information on how the workflow has run. In files, you'll see other available options, like, for example, the Kraken outputs. And in reports, you will see the main information, and you can jump to the different sections, like, for example, the read summary, as I mentioned earlier. The report is interactive, so you can switch between the different samples. And there is also a table that shows you the percentage of unclassified sequences, and also some plots to view the taxonomic profile. So you can explore which is in the sample and identify bacterial, viral, or fungi pathogens. If it's easier to use this Sunburst, it also provides the same type of information, but sometimes this one is easier to explore the lineage. And all the plots can be saved, and also you can do zoom on them. So if you need to compare different samples, you can do it through the bar plot. And here is the abundance table, where for the different samples, you'll see the taxa that has been identified. Also, you can look for the ones that you are interested in. And you can also export the table in case you want to perform further analysis with it. There are also some diversity metrics. And the final section are just the software version and also the workflow parameters that allows you to know which were the parameters that you have used to run the analysis, or in case you want to repeat it, you have this information also in the report. And now I give the floor to Alex, who is gonna explain you how is the workflow testing. Thank you very much.
Thanks, Natalia. Right, now some of you may be interested in looking at what we've done this. What sort of testing have we done, and what can we expect to see when you do this yourself? So here's a glimpse into some of the testing and benchmarking we've done with this workflow, okay? Here are some sequencing metrics. If you're the sort of person that's really into sequencing metrics, this is for you. So what do we expect to see from this? From a MinION flow cell, we expect to see 20 gigs, above 20 gigs data output, which is great to see. So what I want to really highlight from this, actually, the important thing to take away from this is to do with the flexibility of our sample batching, right? What you can see here is we've done some runs with 6 samples, and we've also done some runs with 11 samples. And you can see that actually the yield, the output from the sequencer is not different, right? We've got 25 from 6 samples, 22 from 11 samples. It's very similar. So the yield we're getting is not dependent on the number of samples we're getting. What does change here is the amount of data per barcode we're getting. So if you are having to think about something, it'll be that. Think about how much data you need per barcode, per part of the sample. For example, 11 samples here by 22 gigs. What are we getting? 2 gigs per sample. But remember, because they're on separate barcodes, that's a gig per barcode. So a gig on your viral and acellular alarm, and a gig on your bacterial and fungal alarm. And then we can see some other stats here. If you're really interested in looking at what they are, that's for you. So what do we want to talk about now? Well, I want to show you some of the testing we've done in terms of what we can find. So here's some inclusivity testing. How have we done this? So this is using a Zeptometrics control panel. It's got a load of organisms in here. What did we do with this? So we split this control. You can see there's control 1, which has a load of organisms, and control 2 that has a load of organisms. We split those as a sample each, and we've spiked them into some sputum here. as a normal background that we might expect from a respiratory sample to be doing some pathogen surveillance. Now, if you want to try this yourself, go ahead. You can get this control panel from Zeptometrics, this natural respiratory panel 2.1. Get that, spike it into some sputum, and see what you get. The reason we've chosen this panel is because it's got a really nice mix in it of all the things we want to be able to demonstrate that we can see. There's a DNA virus in here from adenovirus. There's a load of RNA viruses in here. Bacteria, we've got Bordetella in here as a bacteria. But there's also some atypical ones in here, stuff that's going to get really interesting. So chlamydia pneumoniae, for example, that's an intracellular pathogen, and Mycoplasma pneumoniae, that lacks a cell wall. So there's a real mix of organisms in here to demonstrate that we can do it all. And why that's really important in this context is thinking about that depletion at the start, right? So that depletion before the extraction adds process, it adds complexity, and it also affects cells, right? So we're using it to remove certain cells. We're moving it to deplete human cells. We've got to show that that doesn't negatively affect the samples we're putting in. We need to show that once everything's going through the process, we can still find it. So that's why we've done this, because if we don't find everything, we're not really doing a great job of metagenomics and looking for everything, are we? So let's look at what we detected here. This is control panel 1's results. Like I said, they were split. The control panel comes with two different pots with different things in. So control panel 1 here, green is good. That means everything's always detected. And we see those atypicals. We see them both here. They both came through, I want to point this out actually, they both came through the viral arm, those atypicals. That's really important to think about because we've got the acellular one, for example. It's not going to pellet to the bottom at the start when we do that spin. So that's important if you're thinking about only doing one part, right? If you're saying, okay, I'm only interested in bacteria, so I'm only going to do the bacterial arm, okay? I'm only going to spin that one. I'm not interested in viruses, so I'm not doing that arm. Bear in mind, if you are looking or thinking you're looking for something that might be a bit different like that, something acellular, for example, you're going to want to run that viral arm or you risk missing it, okay? Orange here, we've got some variable detection. That means some of the replicates were good, some were missing. In terms of how many replicates were done here, the 6 sample runs here was done in triplicate for each for panel 1 and panel 2. And for the 11 sample run, we've got 5 and 6 replicates here. Now, red is undetected, and this may be due to the currently recommended database, okay, for this metapneumovirus B that we find just no reads for at all. This is done using the 8 gig Kraken database. The 16 gig database is more comprehensive, and we might be expecting to find more things if we'd run it like that. And the other caveat I actually want to point out in this table here is the current version of workflow metagenomics can struggle with subtyping sometimes. That's why flu A's, they're all grouped together in one here. Just that's the reason for that. And then looking at panel 2 here, again, lots of green across the board. We're finding those bacteria, we're finding the RNA viruses. You'll see there's one undetected here, this subtype of a coronavirus, HKU1. Now, I want to note the reason we think that is because in the panel that's provided here, there's only a really short sequence of the genome, a really short sequence of that, and that's why we think we don't detect it, okay? It's the only coronavirus that's missing. All the other coronaviruses you can see are there all the time. Now, how much are we expecting to see here? How sensitive is this? So we've got an LOD experiment here, limit of detection, and this is similar again. This has been spiked into sputum, just using quantitative controls this time. Now, this is our bacterial and fungal slide. So we've got Candida, we've got E. coli, and we've got Staph aureus. These have been spiked into 3 different sputums. Why 3 different sputums? Well, because we know the background can have a significant effect on the sensitivity of the results. Different samples have different amounts of background, so we're going to be depleting different amounts out. That's why we checked against 3 sputums. If you are doing testing of your own here and you want to do this sort of demonstrative approach, I would recommend using more than 1 sample just to check, just to show the difference that backgrounds might have, okay? So our bacteria here, we see we've detected them down to 10 to the 3 CFU per mil, and the fungal one here is down at 10 to the 2 CFU per mil. Now, looking at viruses, we've done the same thing here. DNA viruses, we have detected, that's the adenovirus for the DNA virus, 10 to the 4 viral particles there it's got down to. Flu A, that is an RNA virus, but it's also a segmented RNA virus. That's we found here down at 10 to the 4. And RSV, covering RNA viruses again, that was down at 10 to the 3. So 10 to the 4, 10 to the 3 to 10 to the 4 is what we would expect in terms of how sensitive we are detecting viruses here. And we've got all our bases covered here, right? DNA, RNA, segmented RNA. Now, one thing I spoke about much earlier that I wanted to talk about now was those optimizations that were done to the original Smart9 method and sort of why they were done and what impact we were seeing. So we're looking here at PCR cycles and primer concentration. That's something that was changed, right? So we showed that by increasing both the PCR cycles and the primer concentrations, even though this actually reduced the read length, we were getting better coverage across those segments you can see down here in those dark bars. So that's a trade-off we've done. That's one of the optimizations that's been made to the original Smart9N protocol once it's been brought in here, along with that bacterial fungal saponin-based protocol that's gone for the bacterial and fungal arm. And I'm just going to show you here just what that means in terms of coverage, actually. So flu A, segmented RNA virus, and we can see how much more depth we were getting across the genome here, how much more coverage we're getting. So by increasing that primer concentration, those PCR cycles, and we see here that we're getting 50% of the flu genome is covered at above 20x. And that's in a mixed sample, right? That's a mixed viral sample with all sorts of things in there. In a flu-only sample, we're looking at getting 90% of the genome above 10x. Really good coverage. So this was a lovely thing to see, and one of the reasons we're really putting this forward now, right? Now, what other additional benefits can we get from metagenomics? What have we looked at there? Lineage calling, okay? So this is things you can't do with targeted panels, right? You can't find new things. So one of the things we can look for here is SARS-CoV-2 variants, right? Lineage calling here is fantastically used for, it's just great for pandemic preparedness, surveillance with that sort of things in mind. What can we see? Where can we see things change? How do we see them change? And here's just an example of using it. That's the coverage we've got there on a SARS-CoV-2 genome. We were able to call the lineage from it. Now, let's talk about some considerations, right? What else do we have to think about here? This is a relatively high complexity assay, okay? So realistically, I'm not going to recommend processing more than 6 samples at a time if you're doing both arms manually. Because think about it, each of those samples is going to be split into 2, and then they're going through different processes, not for the whole way, but for the first few steps, there is a different process going on before we get to the PCR stage and they're both together, right? So I wouldn't be trying to do too many of those at once. If you want to do more, and you can be flexible, of course you could prepare some and then prepare more later. If you're in a real rush and need to get things out, we have that flexibility to prepare a few and start sequencing them early. So it's up to what you need to do, but that's a recommendation I'm going to throw in here. We also really have to think about the sensitivity and how it's affected by the background. Like I say, those different sputum samples that I showed a couple of slides back, it's not exactly the same detection we're getting from every sample. That background's having an effect, and we need to think about how that might affect things later on in terms of what our sample is. Firstly, how much is in our background of our sample, because one sample of the same type, one sputum is not going to be the same as the next sputum. They're going to have different amounts of background in them. But also, if we're doing different sample types, you don't have to use sputum for this. Different sample types will have different background. But that really emphasizes, right, the importance of having a really good depletion method, okay? So that's why we're doing this the way we are. So let's summarize now. Let's look back. What is everything we've learned? What are our takeaway messages from today? So in summary, surveillance metagenomics is great. We're going to use it to facilitate comprehensive detection of everything. Bacteria, fungi, and viruses. We want to be able to find it all in one method, and surveillance metagenomics allows us to do that. Using an agnostic approach like this allows us to identify new things. We can find new mutations, new variants, new ways of behavior, all while tracking potential outbreaks as and when they happen. Speaking of as and when, this workflow provides us a rapid turnaround time. We can look at things in real time. We can look at the sequencing data as it's coming off. We can analyze it in real time. And that analysis can be done using the workflow metagenomics bioinformatics pipeline that Natalia talked us through, and you can run that either from the Epitome desktop app, or you can run it through the command line if that's your thing. We've seen that an efficient background depletion is essential for sensitivity. It's so important. That's why this is split into dual arm approach. That's why we're using the depletion methods we are, to be able to find everything. To be able to find everything from one sample, we really have to do this. We have to split into that dual arm. We need to take our sample and split it. And I will stress as well, we're not looking at taking two samples here. This is just, we start with one sample, centrifuge, and we've got both sides. We're not having to take two samples to begin with, so don't worry about that. But we need to treat them slightly different because we're looking for very different things, and this is the approach to do that. And when we do that, and when we get it right, we get detection as low as 10 to the 2 to 10 to the 3 CFU per mil for bacteria and fungi, 10 to the 3 to 10 to the 4 viral particles. And the improvements that have been made here on the original Smart9 method, that has resulted in better detection of RNA viruses, especially short segmented ones such as flu A. So this is the reasons we've done this. This is why these things are so important. Thank you so much for listening to everything we've said. And yeah, if you have any questions, now is the time. Thank you.
So thank you, Alex and Natalia, for those fantastic presentations. You can find more information about today's broadcast using the QR codes, which you can find on your screen. This knowledge exchange, along with all of our previous broadcasts, are available to you to watch on demand. So please do visit the Oxford Nanopore website to check them out. You can also find more information on our upcoming webinars and knowledge exchanges on the website under our events page. And if you do have any further questions about your projects or getting started with nanopore sequencing, please contact support@nanoporetech.com. And thank you all once again, and do enjoy the rest of your day.